feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yong Loo Lin School of Medicine Healthy Longevity webinar. I'm Vincenzo Sorrentino, an assistant professor at the Center for Healthy Longevity, and thank you for joining us. Tonight we are going to have Professor Omar Abudayyeh and Jonathan Gootenberg from Harvard University with us tonight. I'd like to remind you before going into the talks about the Q&A function to send questions. And tonight, we are going to have a student from the Center for Healthy Longevity, Grace K. Lee, which will present their own study entitled Sertindol, an antipsychotic drug as a novel TFEB activator for geroprotection.
Thank you for the kind introduction. As mentioned, I'm Grace K, and we'll be looking at Sertindol, an antipsychotic drug, and explore its potential as a novel TFEB activator to promote longevity. So how do we promote longevity? Logically, we can target the fundamental aging pathways to delay aging. And my project aims to target a primary hallmark of aging, which is disabled macroautophagy. Autophagy is a combination of two Greek words, which translates to self-eating. It is the cell's way of recycling and eliminating cellular material, where a double membrane vesicle surrounds unwanted cellular waste, such as damaged organelles or protein. These vesicles then fuse with the lysosome, where enzymes break down the contents inside. Simply put, these blue vesicles are like a rubbish truck to transport unwanted material to the red lysosome, which acts as an incinerator to degrade them. With age, autophagic activity declines. So this leads to the accumulation of waste products and a decline in cellular health, contributing to the aging process. And this is where TFEB comes in. TFEB is a transcription factor that tackles two arms of the autophagic pathway by increasing the formation of both these autophagosomal vesicles and lysosomes. So with more rubbish trucks and incinerators, autophagy activity is increased. Research has shown that increasing TFEB expression and activation may promote longer health span and lifespan. This includes improvements in lysosomal storage, neurodegenerative, and metabolic disorders. TFEB overexpression also promoted an anti-aging phenotype in worms, meaning that the worms live longer. In summary, these results show that TFEB is a promising target for longevity. With the aim of repurposing drugs to slow aging, we performed a high-throughput screen on almost 3,000 drugs from the FDA-approved and pharmacopoeia drug library. We identified 43 potential geroprotective drugs that could activate TFEB and increase autophagic activity with low cellular toxicity. Upon characterization, we saw that antipsychotic and antidepressant drugs formed the second most prominent group next to anti-cancer drugs. We found this quite interesting, as literature has shown that psychotic conditions exhibit dysregulated autophagy and is also associated with accelerated aging. Hence, we hypothesized that antipsychotics could be repurposed for longevity by consuming it at a lower dose and frequency than clinically recommended. One drug that caught our attention was Sertindol, which is indicated for schizophrenia. It is a second-generation antipsychotic, which means that it has less side effects as compared to first-generation antipsychotics. Moreover, it is non-drowsy and has good oral bioavailability, which means that it can stay in the body for longer to have a longer duration of action. If Sorrentino activates TFEB, we will expect TFEB to go into the nucleus and turn on genes that increase the expression of both the rubbish trucks and incinerators. This was something we were able to verify through fluorescence microscopy. Secondly, we also found that Sorrentino increased productive autophagic flux, which involves increased transportation of waste, but productive because of the active degradation and recycling of cargo. So during this process, these green dots, which represent LC3, associates with the double membrane. As we can see on the right, Sorrentino treatment increases the number of green LC3 dots per cell. More importantly, we also looked at LC3 turnover. This tells us whether there is productive autophagic flux, which involves the active degradation and recycling of cargo. When we use the mixture of ammonium chloride and leupeptin to inhibit lysosomal function and degradation, we actually see a further increase in green LC3 dots. So this suggests that Sorrentino increases LC3 turnover. Sorrentino also significantly increased lifespan in both worms and flies, with the effective dose being higher in flies. These results further strengthen Sorrentino's potential as a geroprotector. And this work is only possible because of my lab members and collaborators who provided the cell lines and performed the lifespan experiments. We cannot wait to show you more promising results in future. Thank you for your attention.
Thanks, Grace, for your interesting and nice talk, and good luck for obtaining the next results in your project. Now, I want to introduce Dr. Omar Abudayyeh and Dr. Jonathan Gootenberg, who are pioneering scientists renowned for their advancements in gene editing and molecular diagnostics. Both serve as PI at the Harvard University, where they co-direct actually the Abu Good Lab, focusing on developing innovative tools for manipulating the DNA, RNA, and cellular states. Their collaborative efforts have significantly advanced the field of CRISPR technology. Notably, they also co-founded Sherlock Biosciences, aiming to harness CRISPR for rapid and accurate diagnostic applications. Their work has been instrumental in creating new platforms for gene therapy and diagnostics, with the potential to transform the treatment of genetic diseases and cancer. In recognition of their contribution, both Abudayyeh and Gootenberg have featured in the Forbes 30 Under 30 list in science and healthcare. Their ongoing research continues to push the boundaries of biotechnology, aiming to develop novel therapeutic strategies for a range of diseases and for aging itself. And the topic of tonight, it will be engineering cellular longevity therapies. Omar and Jonathan, the floor is yours.
Thank you so much for that introduction. We're incredibly excited today to talk about the work from our lab on how we can use these new programmable technologies. We really like to think about how we can leverage programmability in biology. And one fundamental question is biology programmable? If any of you are familiar with this pathway diagram, you would probably say no. But metabolic processes and biology in general, I think actually belay a lot of programmable motifs. And we're very excited about the capability to actually be able to understand these different motifs as they relate to aging, and then engineer and reprogram biology to actually create new therapeutic interventions. So with that, we like to think about how we can actually program our biology to rejuvenate cells and actually rejuvenate our bodies. So we think that there's really three key milestones that we can use to approach the outstanding challenge in aging. One is, how do we actually define aging at the molecular level? Two is how we can understand the general aging mechanisms. And three, how we can actually use this information to predict aging interventions with new advances in artificial intelligence. And so we'll talk about all three of these today. I'll talk about the first and part of the second. Omar will follow up and conclude in the second, and then he'll be able to talk about the third. But at first, we need to really think about how we can quantitatively explore and understand aging. So there is a quick answer to that. We can develop molecular clocks. Now, why do we need molecular clocks? And there's multiple reasons. First, we think about aging as a kind of not exactly a single process. And actually, if you think about each of your different organs ages. And so we need clocks to be able to understand each organ and each tissue. However, we also need mechanistic interpretation. And things like methylation clocks, which are currently the gold standard, are not easily mechanistically interpretable. So we need to be able to build high-resolution molecular clocks to mechanistically interpret these different types of aging processes, as well as screen perturbations at scale. And so we often need to be able to build these complex clocks, and actually cost is a big factor for us, which is why we have actually started to create new technologies. For example, we've been developing a low-cost single-cell sequencing platform in the lab to basically to be able to scalably generate data for these clocks. So it's based off of a split pool-based method where we can take cells and pass them through multiple different barcoded wells to be able to actually sequence millions of cells for under $5,000. And this allows us to scale up to potentially, at some point we hope, billions of cell regime of different cells that we're sequencing. So this drops the price of perturbation screens like Perturb-seq to an accessible amount. It also allows us to do profiling of many different patients at many different ages. So we're leveraging UltiSeq to actually be able to generate different data. And this is just performance of UltiSeq, where you can see that it actually generates great data quality comparable to other split pool-based sequencing methods. Here we actually sequenced human and mouse cells, and we found that we did not get any kind of doublets, that all the analysis we saw was able to separate those two cell types. We also saw that the barcodes in this experiment actually mapped well with multiple different, about thousands of unique cells mapped. So this is also a high quality readout. So we're using these sequencing platforms to define aging at the single cell level. So we have been collecting single cell RNA sequencing traces from hematopoietic stem cells that have been collected from donors via mobilized with Plexifor, and we want to be able to actually use these as a clock to understand age. So here, across 5 different chronological ages, we've actually been able to see that we can develop clocks with fairly high confidence that are able to separate young and old cells. So here you can see very young cells have actually separated from the old cells in the population, but down here we've actually used machine learning to build a clock that can actually take these transcriptomes and predict the age. So we hope that these will be a very easy readout for additional screens. We're currently scaling this across a wide range of donors to understand how we can build this clock and improve its accuracy and generalizability. So these clocks actually provide mechanistic interpretability. So we see things when we look at age related to pathways such as cell adhesion or actin and CDC42 and angiogenin signaling. So all known potential mechanisms involved in hematopoietic stem cell aging. We're very excited about, as these clocks develop, where we can find additional potential hits from them for mechanistic interpretability. But a real critical aspect is how we can use these clocks to actually understand and read out perturbation screens. Because finding out different measurements of aging isn't enough. We need to be able to perturb. So when we develop these clocks, we still don't know if these actual things that we're seeing are mechanistic as they're causing disease, whether they're responsive, as in they're trying to reduce disease, or they're just passengers. They're correlated, but not causative in either way. So because of the inherent correlative property of molecular clocks, these alone cannot nominate new targets. This is why we want to use perturbation-based screening. And so perturbation-based screening allows us to take a wide array of perturbations, such as CRISPR perturbations, where we knock out genes, transcription factors, where we can overexpress different reprogramming transcription factors, or even, as you'll see, secreted peptides, and deliver them to a large population of cells. Each cell receives only a single perturbation, and by doing single-cell RNA sequencing on this population, we can actually read out the molecular age of each single modification we're doing. So this allows us to nominate new pathways for aging drivers. And these diverse perturbations, whether they're CRISPR, gene expression, peptides, will be able to intersect and show us new pathways of potential aging targets that we can then use in animal models to validate. So for example, transcription factor-wide screening can be used to discover modulations of cell fate. So previously, we actually developed a transcription factor atlas library, which contains over 3,500 different transcription factor isoforms. And we can actually develop that library and package it into lentiviruses, which can be transduced at low MOI. We previously transduced them into embryonic stem cells. We can let those transcription factors differentiate or modify the cells, then we can read out their impact with single cell RNA sequencing. And when we previously did this on hematopoietic, on human embryonic stem cells, we were able to discover many different modulators of cell fate. So you can see that these TFs, when dosed on cells, actually created a wide variety of different cell fates. So this provides the opportunity to actually see potentially new ways to reprogram hematopoietic stem cells. We're extending this to additional libraries. So we're very interested in secreted factors. And it's very well known that secreted factors can have large impacts on longevity. For example, exerkines released by the muscles during exercise can actually be trafficked to the brain and have an impact on neurons. So instead of profiling different discrete groups of secreted factors, we have synthesized a library of 5,000 secreted factors covering all 2,000 known secreted factors and 3,000 predicted secreted factors that we can then pool in a lentiviral format and deliver to cells. Critically, they have a tethered peptide on them, which actually allows for activation or inhibition of cells only on their membrane. So there's no spread of these peptides to other cells. So this simultaneously allows us to discover new peptides that can actually affect cell outcomes, as well as discover new receptors and pathways that may have impacts in longevity. So we're very excited about using this pipeline and library as well to understand secreted factors in a wide variety of different diseases, including muscle aging, cardiovascular disease, and progeria. So that's just one other way that we're able to use library profiling to understand disease. Now, how do we actually confirm or validate these hits? And for that, I'll actually pass it over to Omar to talk about our different molecular readouts that we're pursuing in the lab.
Great. So now I'll chat about a little bit about how we actually can sort of validate hits we get from our screens and dive into how we're using all the data coming out of our large scale screening efforts to build AI models that can help accelerate some of this work. And so a lot of the work that we sort of try to do when we run a screen is actually run them through a series of assays akin to a frailty score that people do for mice, but for actual cells. And so we've built a series of different assays across increased cell size, mitochondrial dysfunction, senescence, and DNA damage to actually build a molecular frailty score that's highly correlated to age. And this is just showing some young cells versus old cells, and then purposely created senescent cells where these different markers can actually correlate quite nicely. And now we're trying to do this at scale across hundreds of hits we get across many different cell types for the different screens we're doing. We'll hopefully have more to report on that soon. But as we collect, millions and tens of millions of new cell types with different types of multimodal readouts, including, single cell RNA-seq, there's a question of, can we actually start to build a very predictive virtual cell that can actually make predictions about cells, allow for at-scale target biology, and also drug screening. And so in that vein, we've been really excited about how can AI accelerate basically every stage of drug development, from sort of the early R&D work, uncovering new targets, uncovering new biology, to being able to do sort of in vitro drug screening, in vitro validation of the drug screening, like can you predict tox, can you predict in vivo efficacy, and actually then build that up into models that can predict phase 1, phase 2, and phase 3 outcomes in actual humans and trials. We think each of these stages is actually quite tractable, and we have projects around all three buckets. The virtual cell is what I'll talk about a little bit today, which is around the target biology and drug screening areas. There's kind of a question of what even is a virtual cell. And when you look at all the things that you can do, it actually is not just one thing, but we try to think about how to build foundation models that can basically allow for all these applications on the screen across, how can you build a reference atlas to study, evolution and dynamics of cell states, predict actual perturbations, including drugs, and even build that up into tissue and organismal level predictions as well. And when you actually talk about foundation models for biology. There's also a question of what's your data source. In our case, we've been very focused on single cell RNA-seq because it's very, one, there's already hundreds of millions of cells, and then two, it's the easiest sort of output to scale for the perturbation screens that we actually do. And when we think about why now is the time, it's if you actually look at the past decade and a half of data accumulation, we're starting to get to the point where we're really building, we're at 200 million or so cells for human cell states in RNA-seq, and we're at a point where the sequencing costs and sort of single cell screening costs are decreasing, where you can actually start thinking about doing large scale perturbation screening. We're sort of saturating the observational data sets. There isn't much more than 200 million you can go observe for humans. But what you can do is start perturbing every gene in the genome, knocking it down, turning it up, and doing that across all sorts of cell types, doing in vivo screens. And that can give us, many more cell types, hundreds of millions of more cell types to feed into these models to make them more predictive. And so data really is the key here. If you think about other models like AI models for language, like GPT-4, GPT-3, those were on trillions of words and really large data sets, like 50 terabyte file sizes across the internet. Image models are on billions of images and hundreds of terabytes. And when you look at cell models, we really only have 60 million cells, maybe 200 if you really push, all the diseases and variations that have been sequenced. And it really amounts to a couple terabytes worth of data. And so there's really a big push of how can we get to 1 billion cells, right? How can we scale this by an order of magnitude? One of the ways is that Jonathan talked about earlier is by having a decreased cost of sequencing and sort of our cheap multi-seq preparation method, we're able to actually do much cheaper sequencing that allows us to do this greater scale. And so the idea is, can we take our perturbation libraries, like the transcription factor library, across many different cells types, feed that into our sort of pipeline decreased preparation, and sort of try to build the largest perturbation single cell data set for humans. Once you have that larger data set, there's then the question of, what are the types of predictions you want to make? And so there's two types. One is, you might see a bunch of perturbations in a bunch of cell types, and then you have a new cell type, like a disease cell type, that you want to be able to predict. And then the other is you have a bunch of cells with a few perturbations, and you have novel perturbations you want to predict in those cell types. And both have their own challenges and different types of data that you need to provide. But just to give an example, we have many metrics and evals we've been trying to do, but one of the more common ones is, can we take a perturbation and predict what the differential gene expression will look like? And so SGGen is one of the state-of-the-art models out there, and then our model is shown on the right. And this is, for example, different cell types in the liver. Each row is a cell type, and then we're challenging them with a small molecule. And you can see out of 20 the top 20 differentially expressed genes, we can actually predict anywhere from 75 to 95% of those, whereas the state-of-the-art model gets anywhere from 0 to 25% or so. And then for another experiment where it's different cell lines with a genetic perturbation, like turning on, I think, interferon on them, we can actually still get pretty good prediction of the top 20 genes, about 50% to 70%, whereas the state-of-the-art model does not get that. And so this is a really tough challenge of saying, hey, if I turn this gene off, what happens to the gene expression in the cell? That's a very hard thing to do, but we're making really nice progress, as you can see. This is another way to visualize this. This is another eval where you can project the UMAP of the single cell data. And so the green cells are unperturbed cells, the dark teal ones are perturbed, and then the even darker blue ones are the predicted. And you can see that the teal and blue ones overlap, because we're actually predicting the effects of this perturbation. In this case, I think it's interferon on different primary cells, different immune cells. You can see for each of the cell types, we get pretty nice overlap between the blue and teal, showing that we're actually making a pretty good prediction here. One example of how we can use this for aging. So we took a aging muscle atlas from last year in Nature Aging. It's 8 decades of life to sequencing muscle cells. And we actually simulated thousands of small molecules on the old muscle cells and saw if we could make anything look younger. So the green is the old cells, and the teal over here is the young cells. And then we found one molecule that actually brings the cells halfway back to the young state. This molecule was genistein. And we actually found literature support for this molecule molecule that there seems to be some involvement with skeletal muscle regeneration. And this is actually just a supplement. It's a flavonoid, isoflavonoid that you can buy from soybeans on Amazon for like $60 for a bottle. And I don't know, people sell it for like, immune health, but apparently there could be benefits for your muscle as through our virtual cell predictions. But the idea here is, and there's many other predictions that come out of this type of in silico drug screen, but imagine you could do this at scale across, every small molecule that's ever out there. Even de novo molecules, you do this across every cell type in the human body for every disease, every, aging atlas. And then you also do this for every gene. You can do it for gene knockout, genome-wide knockout screens, overexpression screens, and, hundreds of patient single cell data sets. You can do, screens you could never actually do in the lab because it's hard to grow, liver fibrosis samples from 300 different patients. And so we really think there's this ability to eventually build like, these virtual intervention atlases, like you have virtual aging atlases, you can have virtual disease atlases where we can actually do these types of screens at scale and uncover new biology and potentially new interventions. So we're really excited about scaling up this data, using this in combination with even clinical data like biomarkers, health data, aging clocks, to actually even take the next leap of can we predict drugs that will then have an effect in humans and predict actual phase 1, phase 2, phase 3 trial outcomes and go all the way from in silico cloud experimentation to new drugs. So with that said, this is driven by a lot of different teams and people in the lab and many different funders for our support. And yeah, that's a little snapshot of our aging research.
Thank you, Omar and Jonathan, for the very brilliant and interesting talk. I think it's always beautiful to see how the technology actually can allow us to bring forward, not just trying to test things out, out of our hypothesis, but also generate new ideas by using in silico data sets that are becoming more and more available nowadays, and how to apply the technology to that. So regarding a bit the two parts of your talk, a couple maybe questions, right? Regarding, for instance, the first part of your talk, Jonathan, you mentioned about the fact that you are developing these new types of molecular clocks based on the UltiSec platform, right? So in that sense, yeah, compared to the epigenetic clocks, of course, these are based on RNA, which means they are more interpretable, or perhaps they could provide more actionable interpretation, right? So my question is, you have shown us an example of the hematopoietic stem cells. I was wondering, you have shown some pathways that are driving this, let's say, differences. My question is, have you tried this already? I assume, as you guys have shown on other cell types, and do you get very diverse, let's say, factors that are driving the age differences between cohorts, for instance?
That's a really fantastic question. And we haven't actually completed any clocks on other cell types. We're excited about being able to scale to other cell types. And I believe that we'll have very different mechanisms. The other thing I would say is that the clocks not only are interpretable, of course, but they allow us to do screening with perturbations for a aging phenotype, which is something, if you said I wanted to do a genome-wide screen, I wouldn't be able to understand how those things affect quote unquote aging. These clocks, combined with perturbation sequencing, allow us to do that. And that's, I think, their real dual use. So we're very excited about generating clocks across many different tissues.
And since you're talking about different tissues, different cell types, and we know there's a lot of cellular heterogeneity when, in an aging tissue, for instance. One question I have based on the slide where you were showing, for instance, that you can essentially infect and then perturb different aging cells and then observe the changes. At the moment, my question would be, which kind of aged in vitro cell model actually would you suggest or recommend, right? Because you have the hematopoietic stem cells, but are they the best model for your current research, or have you figured, or would you be able to suggest other ways to, work with aged cells? Because going in vivo, it's always a bit of a difficulty to model, right?
Yeah. I think primary cells from different ages is the best way. You could, of course, explore things like organoid models or more complex models. I think that those, for hematopoietic stem cells, can be a little complex and not scalable. I think it depends on the different tissue that you're trying to model more broadly. For us, primary cells and a lot of these tissues that we can source from diverse patients of different ages makes us the most confident prior to going in vivo, which, of course, we can also do.
And what about, for instance, are you working also, for instance, with age-accelerated conditions? I mean, would those be like progeria conditions? I've seen a few papers, people have done, let's say, papers where they've looked at fibroblasts or other cell lines accessible from patients with progeria, and then they've tried to compare them actually to normal aging, for instance. Do you think there is something that with your clocks, for instance, we could learn differently from what people have done so far?
Yeah, I certainly think that the clocks can have interpretations to progeria or accelerated aging syndromes. And I'd say that we like the different progeria syndromes, Hutchinson-Gilford, Werner, Cockayne, for their ability to screen. We're doing perturbation screening in all those different fibroblasts actually currently to understand causative differences that we can use. I will say that despite their convenience, progeria syndromes, of course, mechanistically aren't aged, right? And even though they phenocopy aging, we have a little bit of better sense of why that is than just general aging. So I'm very hesitant to use them as a drop-in replacement for aging, but I think looking at similarities in them and looking at ways to reverse those sorts of different phenotypes can inform a lot on more general longevity science.
And just getting back to the UTSEC platform, right? So this is something that has been developed in your lab. So is that something that will be of easy, let's say, access to different labs? Is it something that you're going to, let's say, essentially provide, as a sort of service, or something that people can reproduce easily? Or how do you envision, let's say, scaling this up, let's say, for the community?
The hope is that people can reproduce it easily. We're not envisioning providing it as a service for anybody, but as we develop these protocols and different kind of technologies, we want to make them as accessible as possible. So we'll be releasing protocols and other ways to be able to easily do it.
And regarding actually the implementation, right, of let's say identifying, for instance, factors or things that could impact aging in an in vivo system and eventually in humans, right? So we are in the translational program here, so people keep asking us, how do we bring things to the clinic, right? So I think what would be in the programmable biology in general, and it can be your current platform, but in general also the other things you guys are doing in the lab, the biggest challenge is still nowadays to actually bring things from the discovery and the predictions, as you have, for instance, shown in your model, all the way to clinic. Is it the delivery systems, let's say, of the interventions or the, let's say, genetic modifications that you may want to propose based on your systems? Is it the diversity of the tissues? Is it the ethics? What do you think would be the biggest challenges there?
I mean, I think, there's different challenges for different modalities, right? Like if you look at our gene editing work, the biggest challenge there has been, one, they're very expensive to develop and optimize and get working high efficiency and, actual people. It's hard to do delivery well. And so delivery, to most tissues is difficult. And so that's limited most interventions. And I think also, especially with gene editing, the number of people per disease is pretty small. And so that also makes development hard as well. With that said, I think on the aging side, I think for other types of molecules like biologics and small molecules, I think the big problem is just the success rates are just low, right? Like you end up doing phase 2, phase 3 trials, and you just don't see the effect you want to see. And so there's a disconnect between, I think, the preclinical models, whatever it is, your monkey models or your mouse models, and even maybe your phase 1 biomarkers and what you actually see with the biology. And so that's why we're pretty excited about these computational models built on large scale multiomics and multimodal data, because our hope is maybe you can identify where the limitations are, right? If you look at, 100,000 trials and all small molecules and drugs and have really rich models that can represent cells and, gene pathways and, even start to build virtual tissues and virtual animal models, could you actually start to identify, why the drugs fail in the clinic for certain diseases diseases, why some didn't, and actually then start to build models that fill those gaps, right? Like imagine you actually could better in silico screen the biology and understand the biology better. We think we could move the success rate a little bit higher, right? And you actually could see more drugs make it through all the way. But that tends to be, I think, the biggest challenge is it's that we don't always fully understand the biology and the models don't recapitulate, to humans. Like how many times have we, cured, I think, a tumor in a mouse, right? And then you don't see that translate over. So, and then on the third, I think for longevity specifically, I think there just isn't good obviously biomarkers for longevity, right? Like we're building all these protein clocks and all these epigenetic clocks, and the clear connection between those clocks and like specific disease outcomes, I think, is lacking. And this is another place I think some of these AI models we're talking about could be useful, because if you actually could start to connect, let's say you have, we work a lot with the Mass General biobank, you also have the UK biobank or whatever. Imagine you could build models on people that do connect, the blood proteomic signatures to their actual disease outcomes across like hundreds of thousands of people. That would be very powerful, and that would actually move the needle quite a bit for starting to build, disease predictive biomarkers that also have a longevity angle. So, that's some of the stuff we think about. Yeah.
Yeah, and I think it's a great point, because I mean that's essentially a little bit, I would say, the challenge that we are facing also in the field of essentially aging clocks, right? So most of the clocks are based even on the in vitro models or in vivo models that we are using for our research are still based on most of the time detecting age-associated biomarkers, right? So which may predict, I'd say, how they are linked essentially to a disease that is linked to aging, right? So whether we can actually identify biomarkers that are really specifically pointing at aging trajectory is something that people are trying to work on. So perhaps my question would be related to that. Do you think indeed that with your technology and your different utilization and prediction of different cell types, you may be able to actually really generate new biomarkers that are more predictive for actually aging itself rather than for age-associated diseases? Do you have ideas about that?
Yeah, I mean, I think it's a good question. I mean, some of the stuff we're trying to do, I think one of the challenges why these biomarkers haven't been as connected to disease, you see a lot for longevity, and that's easy to measure, but there aren't a lot of good diseased, biobank samples, right? And so we've actually been trying to find better biobanks. Like, for example, there's the biobank of National Brigham is much more enriched in disease.
Right.
Of immunologic disease and patients with those types of sort of indications. And we think that's a really powerful opportunity, but there's no proteomics data, right? And so we're trying to hopefully build the proteomics data out. Because I think, with the UK Biobank, it's a lot of healthy people, so you actually don't see that disease connection. So I think that's one of the things is just like, can we identify where, we can, make those connections in the data, I think is really important. And that's one of the areas we look at. I don't know if Jonathan has another perspective on that.
No, I mean, I think that's completely right. Yeah.
Yeah, so now I think that's indeed, where the challenges lay ahead, right? So I think before going to Sanjay, I perhaps, my personal curiosity now, not necessarily related to the type of science you're doing, but why actually you guys are doing together, right? The science that you're doing, right? And usually, this is a question I get also from many people, right? We always try to establish our labs as individuals, but in your case, you come from the same lab, right, as postdocs, and then you decided essentially to continue your career together. So can you tell us a little bit, out of curiosity, why this choice, and what do you see the advantages, and maybe also what are the challenges,
Yeah, I mean, I think, science is science is a team sport, and it can be a little lonely. I think, both in the sense that you don't have someone to brainstorm with all the time, but also you don't have someone to, push back on you. And so I think kind of the idea that you can work collaboratively as a PI in a very tight-knit environment is very nice. We started working very closely together in grad school. I mean, we've known each other before that, but we both did our graduate studies in Feng Zhang's lab, and it was a very collaborative environment, many co-authored papers. And I think when you do that sort of science, it shows you that you go farther together. Now, it's kind of strange that the structure of kind of academic research, where there's like one PI, there's one tenure track slot, et cetera, really disincentivizes that sort of structure. And we've kind of like pushed back against that. I think a little bit. And it's really been useful because we can provide different concepts. We can bounce ideas off of each other. It's way, I think, more constructive than just like having one PI whose rule is like the rule of law. Yeah.
Yeah, that's very, I mean, I think it's a very inspiring, let's say, message also in the sense that science is becoming more and more complex. And I think it makes sense to try to collaborate rather than isolate ourselves as scientists. So for me, it's actually a very interesting example, yours. And within the center here, we try to maintain a very, let's say, interactive environment. So yeah, I think I hope this serves as a good example also for the scientific community overall. So I think I can hand over now to Sanjay, who is a research fellow from my group at the NUS Center for Healthy Longevity, and is here to moderate the Q&A. So Sanjay, you can, if you have questions from the participants, go ahead.
Hey, thanks for that your talk, and it's very nice and interesting talk. So, if I say like this, most of the technique is based on the engineered proteins. So, how would be the like long-term effect of this organism aging?
So just to confirm, so basically, how do we study the long-term organismal effects of like engineered proteins? Yeah, I mean, I think that's a really fantastic question. It depends exactly what sort of kind of biological mechanisms we're looking at. But broadly, there are these kind of whole organism frailty scores that allow us to look at, essentially 20 odd different parameters of aging. So in that respect, we can look, at things like muscle loss, obviously longevity, like lifespan, inflammatory markers, strength, things even like hair loss or hair color and grooming. And so I think that, that frailty score is a great way to actually map these to an organism. Now, with something like hematopoietic stem cells, of course, there we can also look at known things like infectious disease challenge or skew between lymphoid and myeloid lineages. So I would say that there's obviously several general concepts, but several specific as well.
And yeah, it's a question for the Omar. So in like a few slides, last slide, so he showed the genesis in a drug. Like, what would be the side effect and dose? And they observe any difference on the basis of sex, like male and female?
Was that for me or?
Yeah. It's,
Omar?
anyone can answer. But like you showed like one drug like genistein.
Yeah.
So means like my concern like what's this like dose and side effect, because and have you observed any difference between the like yeah, on the aging perspective, female and male, on the sex based difference?
For genocide specifically, we're still validating that finding for its effect on muscle regeneration. I mean, this is very early. But I think that's a good question of like, can you look at sex-specific differences? Can you look at talks? Those are also things we want to build models for. It's also another question about what's the data source. Like, is the data source a mix of, different people, backgrounds, male and female, etc. I don't think that stuff is well understood for like multi-omic, like foundation models. But something that should be looked at. And I think on the tox side, tox prediction is also a big area of interest that we want to also dive more into. So,
Okay. Yeah.
One last question, Sanjay. Yeah, go ahead.
Okay, sorry. Okay, and you talk about the virtual cells. So, like, how do you strategize this virtual cell in other model systems, like in vivo model systems? What's your strategy?
For model systems, I mean,
you talk about the virtual cells.
yeah, I mean, in general, like experimental model systems, or
Yeah.
like how do we validate the findings?
Yes. How do you validate your findings in model system?
We use a combination of the in vitro sort of aged primary cell models as well as mouse models generally. Although we are thinking about doing a little bit of primate work as well for things that have meaningful clinical biomarkers that could translate over to humans as well. Yeah.
Thanks for your answer. So I'm hand over to Vincenzo again.
Yeah, thank you, Sanjay, for the questions. And thank you again, Jonathan and Omar, for your beautiful talk. Looking forward to the next discoveries in this direction. So thanks again, everyone. And I just want to remind the participants to use the chat function and choose panelists and all attendees option to leave comments and feedback so that everyone can view it. And for the next LT Longevity webinar, it's going to be on March 13, and it's going to be with Professor Raymond Mack, who is an associate professor of radiation oncology, Brigham and Women's Hospital, Dana-Farber Cancer Institute. The topic will be confirmed in next days, but stay tuned for that. And then, let's say, to leave you with a message, as always at the end of our shows, we have a final video that is about learning how CRISPR-Cas9 gene editing technology in a bit catchy and humorous way.
CRISPR-Cas9, bring me a gene, encoding for a specific protein. Make a few snips at this coded locus. You work so well inside a streptococcus. Cas9, I'm so alone without your scissors in my chromosome. Cut me up and do it clear. CRISPR-Cas9, bring me a gene. CRISPR-Cas9, keep me a gene, a virus sequence you've already seen, chopped into bits and stored as genomic, with clustered repeats that are palindromic. Cas9, bind with this code, use it to target infections of old, immunize like a vaccine. CRISPR-Cas9, keep me a gene. CRISPR-Cas9, cut me a gene with a precision that I've never seen. Unzip a strand and interrogate it. Seek out your sequence until you locate it. Cas9, lock into place and do your job as endonuclease. Chop just like a guillotine. CRISPR-Cas9, cut me a gene. Snip, snap. CRISPR-Cas9. CRISPR-Cas9. CRISPR-Cas9, bring me a gene by commandeering my repair routine. A strand to match your severed location for some homologous recombination. Cas9, cheap and precise, rewriting genomes from microbes to mice and soothe the humble human being. CRISPR-Cas9, bring me a gene. CRISPR-Cas9, give us a gene, give us a miracle like the one Nazarene. Cause giving the lame their legs and the blind their sight is if you're for dystrophy and retinitis. With CRISPR-Cas9, why did you fall? Outside our power and inside us all, that really good inside a sea. With this terrible, wonderful power unsettling homes, the door to an ethically metal is every congenital malady better to fit into warrant genetics and better to modify man in the manner of getting a recipe, a mammoth or make a rhetoric. Medical means to eradicate aging or cancer or make a fanatic, a massive epidemic, no weapon to bury a single mosquito, no weapon malaria, send a viral infection to bury a cure to the sense of an HIV carrier. Freedom diseases we're free to work out for nature and accident failed to discover and try to be other than all that we ever have been. Bring us a gene. You unprescribe nucleoprotein.
taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.